All right, get ready, because today we're diving deep into iPSCs.
Induced pluripotent stem cells.
Yeah, iPSCs, those cellular blank slates that hold such incredible potential.
Absolutely.
And we've all heard about it, using lab-grown heart cells to fight heart disease,
Right.
or even neurons to treat neurological disorder.
That's amazing stuff.
But how do scientists actually take those blank slates and guide them into the specific cells that we need?
Well, that's the real question.
That's what we're going to try to figure out today.
It is a really fascinating area of research.
Oh, yeah.
And it's moving so fast.
It is.
What's really mind-blowing to me is the level of control scientists are gaining over these cells.
Oh, yeah.
It's almost like directing an orchestra of molecules to create a specific masterpiece.
I love that analogy, a cellular orchestra. So let's talk about the conductors of this orchestra, the markers like SOX1 or T that act like those signposts, guiding the differentiation process. How do they actually work?
So these markers are like molecular fingerprints, and they indicate which path a cell is taking to become a specialized cell.
Gotcha.
So for instance, SOX1, that's a key player in the development of the nervous system.
Okay.
So if you see SOX1, you know that that cell is on its way to becoming a neuron.
So it's like SOX1 is shouting out, hey, I'm going to be a brain cell.
Exactly.
But what about the big picture?
Okay.
How do scientists even begin to differentiate iPSCs into the wide array of cells that make up our bodies?
Well, it all starts with understanding the three germ layers.
Right.
Ectoderm, mesoderm, and endoderm.
Yeah.
And these are essentially the primary building blocks for all our tissues and organs.
Okay, the germ layers.
Right.
So ectoderm, nervous system, skin,
Yes.
mesoderm, blood, bone, muscle, and
Wow.
endoderm, things like the liver and the pancreas.
That's right.
But how do we get from a pluripotent iPSC to, let's say, a functioning heart cell?
So that's where those specific signaling molecules come into play.
Okay.
Think of them as the instructions that tell the cells which germ layer to follow.
Right.
And ultimately what specialized cell type to become.
Gotcha. Okay, so let's unpack that.
Right.
Let's say we want to create those heart muscle cells, cardiomyocytes that you mentioned earlier.
Okay.
Where do we even start?
So cardiomyocytes arise from the mesoderm lineage.
Okay.
So the first step is to coax those iPSCs down that pathway.
Right.
One of the key players in mesoderm development is this signaling molecule called Wnt.
Went.
Yeah, and it's been shown to be crucial in activating a gene called brachyury,
Okay.
which is like the master switch for mesoderm formation.
So Went is like the messenger delivering that becomes mesoderm message.
Exactly.
And brachyury is like the gene that receives that message
Yes,
and flips the switch.
that's right.
Okay.
Initiating this cascade of changes within the cell that guide it towards mesoderm fate.
Wow. So it's this intricate molecular dance
It is.
with the signaling molecules like Went acting as choreographers,
Right.
directing the cell's every move. And all this happens just by introducing Wnt into the environment where the cells are growing.
That's the beauty of it.
Really?
So scientists have learned to mimic the natural developmental cues that occur in the body, but just controlling the cell's environment.
Wow.
It's like creating a mini embryo in a dish, guiding those cells through the same steps that they would go through during embryonic development.
That's incredible.
It is pretty cool.
So we've nudged our iPSCs down that mesoderm path using Wnt.
Right.
Now what happens next?
Okay.
How do we make sure that those cells don't just stay as generic mesoderm,
Right.
but actually become those specialized cardiomyocytes?
That's where things get even more interesting.
Okay.
So once the cells are committed to that mesoderm lineage, scientists introduce another set of signaling molecules that further refine their fate,
Okay.
guiding them toward becoming heart cells.
Gotcha.
So these molecules, like BMP and active NA,
Uh-huh.
act on specific pathways within the cells,
Okay.
activating the genes responsible for heart development.
So it's like giving them a more detailed roadmap,
Yeah.
narrowing down their destination from mesoderm to cardiomyocyte.
Exactly.
But are these signaling molecules always added one after the other,
That's a good question.
or can they be combined?
It's a bit of both, actually.
Okay.
Sometimes scientists use a sequential approach,
Uh-huh.
adding different signaling molecules at specific times
Right.
to mimic the stages of natural development.
Okay.
Other times, they might combine several signaling molecules together,
Uh-huh.
creating a cocktail that pushes the cells toward the desired fate more efficiently.
Okay.
It really depends on the specific cell type they're trying to create and the protocols that has been optimized for that process.
So it's a constant process of experimentation and refinement,
It is.
figuring out the perfect recipe for each cell type.
That's right.
With all this talk about mesoderm, I'm curious about those other germ layers.
Okay.
What about the ectoderm, which gives rise to the brain and skin? What kind of molecular conductors are guiding those differentiation processes?
So in the case of ectoderm differentiation, one of the key players is this signaling molecule called retinoic acid.
Retinoic acid, that sounds familiar.
Yeah.
Isn't that also important for vision?
Yes.
Like, isn't it related to vitamin A?
You're absolutely right.
Good.
Retinoic acid is a derivative of vitamin A,
Mm-hmm.
and it plays a critical role in both vision and neural development.
So the same molecule can have these really diverse roles in the body,
Yes.
highlighting how interconnected different biological processes are.
That's right.
It's incredible how something as seemingly simple as vitamin A
Right.
can be so crucial for such complex processes.
Absolutely.
And I'm assuming there are other key players involved in ectoderm differentiation besides retinoic acid?
Of course, there's a whole network of signaling molecules and transcription factors that work together
Okay.
to orchestrate this intricate process.
Right.
One important factor is this protein called sonic hedgehog,
Oh, yeah, I've heard of that one.
or SH for short. And it's involved in patterning the developing nervous system,
Great.
helping to establish the different regions of the brain and spinal cord.
It's like a secret code guiding the development of the nervous system.
Uh-huh.
And I remember reading about another marker, PX6, that's
Yeah.
involved in eye development.
PX6 is a fascinating transcription factor,
Yeah.
and it plays a critical role in the formation of the eye.
Wow.
It's so essential that mutations in the PX6 gene can lead to various eye defects,
Oh, wow.
highlighting its importance in this process.
It's like PX6 is the architect of the eye,
That's a great way to put it.
making sure that all the different parts come together correctly.
Yeah, and just like with mesoderm differentiation, scientists can use these signaling molecules and transcription factors to direct iPSCs towards specific ectodermal fates,
Okay.
whether it's generating neurons for studying neurological disorders or creating skin cells for potential skin grafts.
So we've covered mesoderm and ectoderm, which leaves us with endoderm. Yeah.
The germ layer that gives rise to organs like the liver, pancreas, and lungs.
That's right.
What are some of the key players guiding endoderm differentiation?
So one of the most important signaling pathways in endoderm development
Okay.
is the active nodal pathway.
Okay.
These signaling molecules are crucial for inducing endoderm formation and directing the cells towards different endodermal fates.
So activin and nodal are like the trailblazers, marking the path towards endoderm.
Yeah, that's a good way to think about it.
And then how do we get from generic endoderm to, say, a pancreatic cell that could potentially be used to treat diabetes?
So that's where things get really specific.
Okay.
So to generate pancreatic cells, scientists might introduce additional signaling molecules like FGF and retinoic acid, which we talked about earlier.
Oh, wow. So it comes back again.
Yeah.
Okay.
These factors help to further refine the cell's fate,
Okay.
guiding them towards a pancreatic lineage.
So it's like this multi-step process.
Right. With each signaling molecule acting like a checkpoint, guiding the cells closer and closer to their final destination.
That's a good way to put it.
And it sounds like there's a lot of overlap between the signaling pathways involved in different germ layers.
That's right.
We've already seen retinoic acid playing a role in both ectoderm and endoderm differentiation.
That's an important observation.
Yeah.
These signaling pathways are often interconnected and can have different effects depending on the context.
Right.
So, for example, retinoic acid can promote neural development in ectoderm, but it can also be involved in pancreas development in endoderm.
Wow.
It's all about the timing, the concentration, and the combination of different signals that ultimately shape the cell's fate.
So it's this incredibly complex symphony of molecular signals that directs iPSC differentiation,
Absolutely.
with each molecule playing its part at just the right time and in just the right way.
That's amazing.
And this level of precision and control is what makes iPSC technology so revolutionary, right? By understanding these signaling pathways and how they interact, scientists can precisely manipulate iPSCs to create a wide range of cell types, opening up this world of possibilities for disease modeling, drug discovery, and regenerative medicine.
Exactly.
It's like having a cellular toolbox at our disposal, with the potential to repair and regenerate damaged tissues and organs.
Incredible.
But I imagine there are still challenges in translating this incredible technology into real-world therapies.
You're right.
Yeah.
While iPSC technology holds immense promise, there were still hurdles to overcome.
Okay.
For example, one of the biggest challenges is ensuring the safety and efficacy of iPSC-derived cells for transplantation.
Right.
We need to make sure that these cells are pure, functional, and don't pose any risk of tumor formation or other adverse effects.
So it's not just about creating the right cells,
Right.
but also making sure that they're safe and effective for clinical use.
That's right.
It's a whole new level of complexity, and I'm eager to explore those challenges further.
Absolutely. There's a lot to discuss.
Yeah, there is.
From the intricacies of quality control and cell purification to the ethical considerations of using iPSCs in regenerative medicine.
Right.
And of course, we can't forget the incredible advancements already being made in using iPSCs to model diseases and develop new therapies.
Of course.
There's so much to uncover.
Yeah.
And we're just getting started.
We're just getting started. Welcome back. We've been talking about this amazing cellular toolbox that iPSCs offer.
Right.
But I'm curious how scientists are actually using them to understand and potentially treat diseases.
So one of the most powerful applications of iPSCs is in disease modeling.
Oh, really?
Yeah, we can literally recreate a disease in a dish.
Wow.
Using cells from a patient with a specific condition.
So instead of just studying a disease in a general sense, you're looking at it from the perspective of an individual patient.
Exactly.
And their unique cells.
Precisely. It's like having a personalized disease avatar.
Oh.
Allowing researchers to study the disease process in a way that wasn't possible before.
Okay, so can you give us a specific example of how this works?
Okay, so let's say we have a patient with a rare form of heart disease.
Okay.
Caused by a specific genetic mutation.
Right.
Scientists can take a small skin biopsy from the patient, generate iPSCs from those cells,
right,
and then use the techniques we discussed earlier to differentiate those iPSCs into cardiomyocytes, the heart muscle cells.
So they're basically creating heart cells that carry the same genetic mutation as the patient.
Exactly. And now they have a dish full of heart cells.
Wow.
That exhibit the same disease characteristics as the patient.
Okay.
This allows researchers to study how that mutation affects the heart cell's function.
Right.
Look for potential drug targets, and even test new therapies in a controlled environment.
It's like having a personalized testing ground for new treatments.
Exactly.
Are there any examples of this actually leading to new discoveries or therapies?
There are some incredible examples emerging. For instance, researchers have used iPSC-derived neurons from patients with Alzheimer's disease to study how the disease progresses, and to screen potential drugs that might slow or halt that progression.
So instead of testing drugs on a generic model of Alzheimer's, they're testing them on cells that actually carry the genetic and cellular hallmarks of the disease.
That's right. It's a much more targeted and personalized approach to drug discovery.
Wow. It's like a whole new level of precision medicine.
It is.
But I imagine there are challenges in using these iPSC-derived cells for disease modeling.
Sure.
How do you ensure that the cells in the dish truly reflect what's happening in the patient's body?
That's a great question, and it's something that researchers are very mindful of.
Okay.
There are always going to be differences between a cell culture dish and the complex environment of the human body.
Right.
However, scientists are constantly refining their techniques and developing more sophisticated models that better mimic the in vivo conditions.
So it's an ongoing process of improvement and validation.
Yeah.
Making sure these models are as accurate and representative as possible.
Exactly. And it's not just about the cells themselves.
Okay.
Researchers are also developing ways to create more complex 3D models.
Oh, wow.
Incorporating different cell types and even microfluidic systems that mimic the flow of blood and other fluids in the body.
It's like building a mini organ in a dish.
That's a good way to put it.
And that level of sophistication must be crucial for studying diseases that affect multiple organs or systems.
Absolutely. For example, researchers studying diabetes are using iPSCs to create models that include not just pancreatic beta cells, which are responsible for producing insulin, but also other cell types that play a role in glucose regulation, like liver cells and muscle cells.
So they're recreating the entire metabolic network involved in diabetes.
Exactly.
Allowing them to study how different cell types interact and how the disease affects the system as a whole.
Precisely. It's a much more holistic approach to disease modeling.
Right.
And it's providing valuable insights into the complex interplay of factors that contribute to diabetes.
This is truly mind-blowing stuff.
It is.
But what about the therapeutic potential of iPSCs?
Oh, okay.
We've talked a lot about disease modeling,
Right.
but what about actually using these cells to treat patients?
Well, the therapeutic potential of iPSCs is immense.
Okay.
And it's one of the driving forces behind this entire field of research.
Sure.
The idea is to use iPSC-derived cells to repair or replace damaged tissues and organs,
Right.
offering new hope for patients with currently incurable conditions.
So instead of just studying diseases, you're aiming to cure them using these lab-grown cells.
That's the ultimate goal.
Wow.
And while we're still in the early stages of translating this technology into clinical therapies, there have already been some remarkable breakthroughs.
I remember reading about the successful treatment of age-related macular degeneration,
AMD?
AMD, using iPSC-derived retinal pigment epithelial cells.
RPE cells.
Yeah, that was a huge milestone.
It was indeed.
It really was.
It marked a major turning point in the field, demonstrating the feasibility and potential of iPSC-based therapies.
So can you remind us how that treatment works?
Sure. So in AMD, the RPE cells, which are essential for maintaining the health of the retina, gradually degenerate,
Okay.
leading to vision loss.
Right.
Researchers in Japan used iPSCs to generate healthy RPE cells and then transplanted them into patients with AMD,
Wow.
resulting in significant improvements in vision for some patients.
So they essentially replaced the damaged cells with healthy lab-grown ones, restoring the retina's function. That's incredible.
It's pretty amazing.
Are there other iPSC-based therapies in development?
Oh, absolutely.
Really?
There are numerous clinical trials underway exploring the potential of iPSCs to treat a wide range of conditions,
Like what?
from spinal cord injuries to heart disease to Parkinson's disease.
Wow, it seems like the possibilities are endless.
They really are.
But I imagine there are significant challenges in developing these therapies.
Of course.
We touched on the safety aspect earlier.
Right.
But what are some other hurdles?
Well, one of the biggest challenges
Okay.
is scaling up the production of iPSC-derived cells
Right.
to meet the needs of clinical trials and eventually widespread therapeutic use.
Okay.
We need to develop efficient and cost-effective methods for generating large quantities of high-quality cells.
So it's not just about proving that a therapy works,
Right.
but also making sure that it's feasible to produce those cells on a large scale.
Exactly. It's a complex manufacturing challenge.
Yeah.
But there are companies and research groups dedicated to developing scalable and automated platforms for iPSC production and differentiation.
It's like taking this groundbreaking technology from the lab bench to the factory floor.
That's a good way to put it.
And I imagine there are also regulatory challenges involved in bringing these therapies to market.
Of course, regulatory agencies like the FDA in the United States play a crucial role in ensuring the safety and efficacy of any new therapy.
Right.
iPSC-based therapies are no exception,
Okay.
and they undergo rigorous testing and scrutiny before they can be approved for clinical use.
It's a long and complex process, but it's essential for ensuring that these potentially life-saving treatments are safe and effective for patients.
Absolutely. And as the field continues to advance,
Yeah.
we're learning more and more about how to best characterize and standardize these cells,
Okay.
making it easier for regulators to evaluate their safety and efficacy.
It's amazing to see how quickly this field is evolving, from basic research to clinical trials to hopefully widespread therapeutic applications.
It's an incredibly exciting time to be involved in iPSC research.
It really is.
The pace of discovery is accelerating,
Yeah.
and the potential to transform medicine is truly remarkable.
I'm curious, with all the potential applications of iPSCs,
Okay.
from disease modeling to drug discovery to cell therapy, what are some of the most exciting areas of research right now?
That's a great question.
Where do you see the field heading in the next few years?
There are so many exciting avenues being explored right now.
I bet.
It's hard to pick just a few,
Right.
but one area that's really capturing the imaginations of researchers is the potential of iPSCs to create organoids.
Organoids.
Yeah.
What are those?
These are tiny self-organizing structures that mimic the function of actual organs.
So we're not just talking about creating individual cell types anymore.
No.
We're talking about building mini organs in a dish.
That's right. Think about it, a mini liver,
Wow.
a mini brain, a mini kidney,
Huh.
all grown from a patient's own cells. The possibilities are truly mind-boggling.
Okay. I can already see how this would be incredibly valuable for disease modeling and drug testing.
Absolutely.
But could you actually use these organoids to replace damaged organs in a patient?
That's the ultimate goal.
Wow.
but it's still quite a ways off.
Okay.
The challenge is to create organoids that are complex and functional enough to truly mimic the intricate workings of a real organ. But the progress being made is astounding.
So can you give us some examples of the amazing things that researchers are doing with organoids?
Absolutely.
Yeah.
Researchers have created brain organoids that exhibit electrical activity.
Wow.
Similar to a real brain, they've grown mini guts
Mm-hmm.
that can absorb nutrients, and even mini lungs
Oh, wow.
that can expand and contract, mimicking breathing.
It's like science fiction becoming reality.
It really is.
What are some of the diseases that researchers are targeting with this organoid technology?
Well, one area where organoids are already making a big impact is in cystic fibrosis research.
Okay.
Cystic fibrosis is a genetic disease that affects the lungs and other organs.
Right.
And researchers are using iPSC-derived lung organoids
Okay.
to study how the disease progresses and to test new drugs that might correct the underlying cellular defects.
So they're essentially recreating the disease in a dish using cells from patients with cystic fibrosis.
Precisely, and this allows them to study the disease in a much more personalized and targeted way than ever before.
It's incredible how iPSC technology is giving researchers this unprecedented level of control over cells and tissues.
It is.
But I imagine there are ethical considerations that come along with this level of power.
You're absolutely right. As with any powerful technology,
Yeah,
it's crucial to use it responsibly and ethically.
of course.
For example, with brain organoids, there are questions about consciousness and sentience.
Right?
How complex can we allow these organoids to become before we start crossing ethical boundaries?
It's a profound question.
It is.
It's almost like we're venturing into uncharted territory,
Yeah.
where the lines between science fiction and reality are blurring.
It's an exciting but also humbling time in science.
It really is.
We're pushing the limits of our understanding of biology and our ability to manipulate living systems.
Right.
And with that comes a responsibility to ensure that we're using this knowledge wisely
Of course.
and for the benefit of all humankind.
Well said. So we've covered a lot of ground in this deep dive into the world of iPSCs. We've gone from the basics of cellular differentiation to the intricacies of disease modeling
Right.
to the mind-blowing potential of organoids.
It's amazing stuff.
It's clear that iPSC technology is revolutionizing medicine and biology as we know it.
It's been an incredible journey exploring this fascinating field with you.
It really has.
And I think it's safe to say that we're only scratching the surface of what's possible with iPSCs.
Yeah,
The future of medicine is being shaped right before our eyes.
it is.
And it's an exhilarating time to be witnessing these breakthroughs.
It is. Before we wrap up, I want to leave our listeners with one final thought. Imagine a world where personalized medicine is the norm,
Okay.
where diseases can be modeled and treated using cells derived from our own bodies,
Right.
and where damaged organs can be repaired or even replaced with lab-grown ones.
It's an incredible thought.
That's the world that iPSC technology is helping to create, a world where the seemingly impossible is becoming possible.
It's a future filled with hope and possibility.
It is.
And it's a future that we're all a part of creating.
Thank you for joining us on this incredible deep dive into the world of iPSCs.
It was my pleasure.
We hope you've learned something new and are as inspired as we are by the potential of this groundbreaking technology to transform human health and well-being.